Tobias Pusch. Dr. Pusch is an infectious disease physician here at Providence. Among his other duties, Dr. Pusch is one of the physicians in the Providence St. Vincent Travelers Clinic, which in different times is there to help you as you prepare for safe international travel. So hopefully we'll also be being able to take advantage of those services that he offers. And in addition, early in the pandemic, Dr. Pusch was one of the lead clinical investigators in the remdesivir trial here at St. Vincent, which was testing the effectiveness of the COVID treatment. So today, Dr. Pusch is going to be talking with us about all things COVID vaccine. So now, Dr. Pusch, I'll turn it over to you, but welcome and thank you for joining us today.
Kevin, thanks for the invite. I appreciate that. It's always fun to talk about vaccines and infectious disease in general. And that was a very good intro to the meeting here, I think, today with showing the video clips. I've gotten my first shot and I'm looking forward to getting the second one. And, you know, I've had zero side effects, same for my colleagues. And I think that's the overwhelming experience in most healthcare workers so far. Kevin asked me to be not too technical about the vaccines. So some of the sort of bioengineering slides I'm going to be relatively brief about, and I've put in several sort of FAQ slides in. Hopefully they will address some of the questions that have been posed already. My PowerPoint will not allow me to look at the messages in the chat during the talk. So Kevin, if you could monitor that and sort of compile them as you seem fit, and we can address them then afterwards. Okay, so I hope we'll have enough time afterwards for questions, so I will answer those to the best of my ability. So I'll go ahead and share my slides here. So I think you should be able to see my main slide deck now. Although, one second, let me turn off the subtitles here. So some of those I presented during a town hall meeting a couple of weeks ago after the emergency use authorization was came in for the Pfizer vaccine. I retitled this into coronavirus vaccines because obviously there's several now and there's different stages of approval, but we'll go through the different types of vaccines, and then generally talk about what the status is of the vaccine administration at this point. So there's a lot of different types of vaccine platforms. The one that you've heard most about is the so-called genetic vaccines, and we're largely talking about mRNA vaccines. There are two vaccines from Pfizer and from Moderna that were just approved. There's a whole lot more mRNA vaccine. On top there, on top of the vaccine listing there, it says CureVac. I'm from Germany, so there's another German mRNA vaccine that's also now in phase 3 clinical trials. This particular slide here that I threw in is from October that I made for a pharmacy therapeutics meeting presentation. And so these vaccines have moved on in their phases in the interim, but there are mRNA vaccines, and then there are so-called DNA vaccines. All of those are lumped into these genetic vaccines. But there's other efforts. So there's viral vector vaccines, and that's the most recent one that you've heard about, the AstraZeneca vaccine, which was just approved in the UK just days ago. And that falls into viral vectors, where a virus essentially is used where the genetic components of the original virus has been removed and replaced by the target genetic material that we want to have expressed and have the immunity being developed against. Then there's so-called protein-based vaccines, which have not really played yet a role, and there's very little data yet available about how effective they will be. And the other large component is the so-called inactivated or attenuated vaccines, which we use quite a bit in general vaccine clinic. And those also have come to attention now because in India, the so-called Bharat biotech vaccine, which is an inactivated vaccine, has just been approved in India for use. The New York Times actually has a vaccine tracker on their website, which is rather phenomenal. And it's actually not too scientific, too medical lingo containing site. It's very worthwhile reviewing if you have not seen that as of yet. You can just literally Google it and get to the site, and it's very frequently updated. It's quite useful. So we're largely talking about the mRNA vaccines because those are the two vaccines that are in use now in the United States. The BioNTech vaccine, the Pfizer-BioNTech vaccines, and the Moderna vaccines, both of which are mRNA vaccines. So what are mRNA vaccines? We'll get to that in a second once I show you sort of the timeline of this, because it's absolutely unprecedented. And for the medical scientific community, this is frankly astounding. And, you know, when you wait for a vaccine like the Shingrix vaccine for varicella-zoster, which took years and years to get into clinical practice, what's happening in 2020 was truly unbelievable. So the genetic code for the virus, the COVID-19, the coronavirus, was identified and published by Chinese researchers in the Wuhan Institute in early January of 2020. And the response by the BioNTech company in Germany was to essentially use the existing platform of delivering mRNA, messenger RNA, into the body for the coronavirus. So they started working on this as soon as the genetic sequence was released. They partnered then with Pfizer, which started 2 months later because BioNTech is not that big of a company, and to doing large-scale clinical trials, they needed essentially a company that would be able to facilitate that. They were able to launch phase 1 clinical trials by May of 2020. And soon thereafter, once those were completed, the phase 1 trials in end of July, they started conducting larger efficacy trials, and then eventually phase 3 trials, the most important trials that we do in clinical practice. And those are worldwide trials. Most of the trial sites were in the United States, but it was truly an international trial with sites in Germany, in Brazil, and Argentina. The initially enrollment goal was 30,000. A little bit later, the Pfizer CEO announced that they would increase this to 44,000 participants, and that was a completely non-political decision. This was based on increasing power and having enough meaningful results to have confidence about using the vaccine. So by November 8th, speedy progress in the trial. The first 94 cases of COVID were released for initial analysis, interim analyses, and those were then reviewed by the FDA on December 8th. And the trial results allowed a conclusion that the vaccine had a 95% efficacy. And all of that led to the EUA, which was granted on December 11th. And we've been using that, including at the Providence system, ever since. So what is messenger RNA? Well, it's a modified RNA. RNAs are ribonucleic acids. They play a lot of roles in the body in the cell biology. Messenger RNA typically travels from the nucleus to protein machinery stations in the cell, gets translated into the particular product, the protein that the body needs. So this particular RNA encodes the genetic sequence of the spike protein of the virus. There's modifications to it made in a way that it can easily be expressed on the cell surface, and the body develops an immune response to it. So the early trials from phase 1, they were showing that there was a rather fabulous and overwhelmingly positive immune system response to the vaccine. And now we have the phase 2 and 3 clinical trial evidence that this is confirmed in clinical practice. So you may have heard about the delivery systems that it uses. Why do we have this? Why don't we just inject this messenger RNA into the body so the body can develop the immune response against it? Well, there are several reasons for it. So the messenger RNA is not a very stable product. It is normal for the body to have enzymes to degrade these structures, these RNAs, and they wouldn't last really any significant time for the immune system stimulation at all. In fact, there's estimates if you don't have a lipid nanoparticle where it's basically embedded into, that only 1 in about 10,000 molecules would be picked up by cells, and potentially then the immune system reaction would ensue. So there's multiple different components to this lipid nanoparticle, and it's not important exactly to know the exact composition, but picture it, there's sugar molecules on top that help prevention of immune destruction by this early molecule. Then there is surface protein particles to it that allows it to be taken up by exactly the cells that are supposed to produce these protein structures on the cell surface for them to be presented to the immune system. So there is lots of modifications to it. And as I said, I don't want to be too technical about it, but every step of the way that the cell usually uses to modify a protein is basically also conducted here with this synthetically modified genetic product. So which cells are utilized in order to uptake the vaccine particle, then utilize the messenger RNA for it to be translated into a protein that's being presented at the cell surface? Those are usually your muscle cells. So when you get an injection into the deltoid muscle, your myocytes, the muscle cells, will pick this up and present this to the surrounding immune cells, that are present everywhere in the body. There's other very important cells, which is important to mention. They're called dendritic cells. Dendritic cells are early recognizers of foreign invaders, so to speak, and they present foreign proteins really well. And that's also very important. So those are the two key cells that do present the protein, the spike protein of the virus to our immune system. So what are the big advantages of these mRNA vaccines? Well, it's the potency. You really need a tiny, tiny little dose. In the Pfizer vaccine, it's 30 micrograms, which is unheard of in minuscule dose. So the other thing, and that's really important when you think about attenuated vaccines or live vaccines like the yellow fever vaccine, for instance. The yellow fever vaccine is really problematic because we can't give it to immunocompromised individuals. You can literally get the infection from the vaccine. That is not the case here at all. This is a non-infectious, this is just a small piece of a genetic product that is not integrating into our own DNA. So the half-life is relatively short and it's precisely regulated. So the lipid nanoparticle, every component of this vaccine is engineered to last an exact specified, engineer-specified time frame. It's not a genetic vector like the adenovirus vaccine, for instance, from AstraZeneca. So anti-vector immunity, which is a concern with the AstraZeneca vaccine, is not really a problem here and has not turned out to be a problem as of yet in the trials of the mRNA vaccines. So,
Dr. Pusch, I'm sorry to interrupt. Yes. We may have a question. Barbara, are you trying to ask a question? Maybe not. Sorry about that. Please continue.
No problem. Yes. So the vaccine is degraded by normal physiologic processes. There's nothing unusual about it because it's just a normal genetic product injected and degraded. The development that's very important for addressing the pandemic in a timely fashion. These are PCR polymerase chain reaction, meaning rapid production of genetic sequences. This is on a logarithmic scale. These products are developed and can be embedded in these lipid nanoparticles very fast. Nothing like routine vaccine processes from other known vaccine platforms. As you may have heard, the Pfizer vaccine, there's supposed to be over 1 billion produced by the end of 2021. For Moderna, is similar to about 1 billion. It's still, of course, only a fraction of the world's population, but thinking about usual production of vaccines, this is astronomical levels. The cost is low. The US government pays about, I think, $18 or $19 per vaccine compared to many other vaccines. Indeed, this is a very, very low price. This is a vaccine platform. So if anything changes in terms of the genetic code of the virus, which now is in the press, and I have a couple of slides in this slide deck as well regards to genetic variants. We could re-engineer the product, change the sequence of the mRNA, and simply embed that in a lipid nanoparticle and inject that. So it's very easily modifiable. The administration has been safe, is a simple vaccine shot as you would anticipate, and that's been extremely low-key problem thus far from the trials as well as the real-world data. So this is essentially a perfect platform for newly emerging pathogens because there's rapid response possible. So what are the safety considerations just from a manufacturing perspective? So there's no toxic chemicals in it. There's no cell cultures involved, which is really important because live viruses, viral vectors, or inactivated virus contamination is virtually non-existent. The short manufacturing time, that may not be as important if you sort of think as a layperson about it, but actually the longer it takes to produce a vaccine, the more possibility there is of bacterial contamination of vials, and that's been widely reported in the medical literature. And there's no genomic integration, as I mentioned. So how did these vaccines perform in clinical trials? You've heard a lot about these days, and I don't want to be too extensive about it, but I think it's worth going over a couple of slides. So the Pfizer vaccine was injected to individuals 16 years of age and older. There's two vaccines now given. Two vaccine shots at day 0 and day 21 about. There's a leeway of about 4 days, 17 to 21 days window after the first it can be given. And the primary outcome was safety and symptomatic COVID-19. So in the Pfizer vaccine group, there were 8 symptomatic COVID-19 cases. In the placebo group, 162, and that's measured from 7 days after the second vaccine shot was given. So that's how the 95% efficacy of the vaccine came into being in terms of calculation. There was one severe case of COVID-19, severe is being defined, hospitalized with hypoxia. There were 9 in the placebo group. This is the slide from the New England Journal of Medicine from the publication last month, which is nice to have kind of a photographic sort of impact of this. My 11-year-old daughter could interpret this. You see the vaccine arm as depicted in red, and the placebo arm in blue. In every square or circle on the vaccine area denotes one particular case of cases of COVID. For the placebo, actually more than one. What you have in the little window up there is more detailed. We're roughly at 10 or 12 days after vaccination of the first shot. You seem to have actually impact where the immune stimulation from the vaccine sets in, and that's where the curves divide. On the bottom, you see the actual caseloads depicted. So this was a randomized and double-blind placebo-controlled trial. And you see here the inclusion and exclusion. So noteworthy, children and adolescents below age 16 were not included. People by history of COVID-19 were also not included in the trials. Immunosuppressed individuals in any shape or form were not included, and pregnant individuals were not. You could have a stable medical condition of any kind, pulmonary, cardiovascular disease, etc., but otherwise people were largely healthy. In the Moderna trial, there were about 22% of individuals had some sort of underlying medical condition, but the vast majority were healthy individuals. So in terms of the safety outcomes, this is from the Pfizer trial. You see on the left people below 55 years of age, and on the right, greater than 55 years of age. The reactions to the vaccines in terms of local reactions as well as fatigue or headache, the most common reactions after the first dose. Interestingly, the older population had less problems in terms of those side effects, which usually is a factor of immune senescence, meaning the older we get, the less active our immune system is, the more sluggish it is, the more problems occur. Herpes zoster is yet another example, occurs much more frequently obviously, the older we get. Severe side effects were rare. So, and you see, both placebo and the vaccine groups had side effects. Severe meaning in any category. Headache, they were so severe that they had to be treated in the emergency department. Diarrhea to a point that, again, you had to be treated in the emergency department, or you needed admission for any of those measured side effects.
Dr. Pusch, in the
Yes.
vaccinations that are taking place with the Providence caregivers, are you seeing similar results, or is the data not yet available?
There's no clear data available. And, you know, quite frankly, we as a healthcare system don't take sort of note or monitor this closely. How many people develop headache? How many people develop a fever? You sign up to a so-called vSafe, vaccine safety monitoring tool from the CDC, and you will get a daily messaging through a regular phone message system where you will record if you have fever, if you have headache, if you have diarrhea, etc. It's voluntarily, but I certainly would encourage you to do so, because the vaccine, those safety data will be published by the CDC. And, you know, it's already been close to, I think, 3 or 4 million vaccine administrations. So the CDC, I'm sure, will analyze those data soon and make them publicly available in regards to what the large impact has been, not just from the trial data. So we within Providence have only looked at true anaphylaxis-like reactions, and there were a few individuals that had to be treated in the emergency department, but not hospitalized, that I'm aware of. But otherwise, we don't take, you know, that close of data collection within the system regards these different outcomes. So noteworthy here, what I singled out was, in particular, fever, which you see here on top after the first dose and on the bottom after the second dose, which is important because you have about 4-fold increase of fever that occurs after the second shot, which is a booster. The immune system is primed. You have a more vigorous response, and that you see in any of the different side effects that are being assessed here. But in particularly, you know, in the healthcare community, healthcare provider community, this is relevant because we vaccinate sometimes, you know, people from different in the same unit. Yeah, if you have, let's say, in a cardiology ward, you vaccinate a bunch of people, and a lot of them get fevers. Those would be anticipated to be out of work duties for a couple of days theoretically. So scheduling people at different times has been done for us at Providence St. Vincent's. The vaccine side effects have been largely sort of muscle pain, headache, fatigue, which you generally anticipate, just like with the influenza vaccine. But things like shortness of breath, cough have really not been problems with the vaccine. This is all from the trial, obviously. So other safety considerations, both side effects, vaccine and both placebo recipients were very mild and moderate. Expected transient injection site reactions were really only after the first 2 to 3 days. You know, not long lasting whatsoever. The, you know, widely quoted side effects of anosmia, meaning you're not able to smell, and ageusia, not able to taste, were related to the infection itself, not seen with the vaccine. And again, severe vaccine reactions were really uncommon. The Moderna trial was a bit smaller than the Pfizer vaccine trial that included almost 45,000 individuals. Otherwise, the trial was fairly similar. The administration of doses of 100 micrograms, so about 3 times the dose, was on day 0 and day 28. The primary endpoint, same, symptomatic COVID disease. So for this trial, there were 185 individuals who developed COVID-19 illness. COVID-19 illness being defined as having PCR positive individual with at least one symptom, which could be a variety of things, shortness of breath, fever, cough, or a multitude of symptoms. And here you had 11 cases and no severe cases that were severe, which is really important because not only want to we want to prevent COVID, period, but also hospitalizations to, reduce the burden on the healthcare system. This is also from the New England Journal paper that was just published on December 30th. And you see a similar, it essentially matches the Pfizer vaccine trial. On top there, you see the incidence rate that's listed per person, per 1,000 person years. And so there is a reduction about 14-fold from about 18 cases in the placebo group per 1,000 person years to only about 5.6, which is quite dramatic. And there's a summary on the bottom of how many cases you have in each group. The Moderna trial started counting cases by primary endpoint 14 days after the second dose, slightly different than the Pfizer vaccine trial. I said I would say a couple of words on the AstraZeneca Oxford vaccine, which is a very different platform, very noteworthy. So this is a viral vaccine, vector vaccine class, has nothing to do with genetic vaccines. So what it's utilizing is a chimpanzee adenovirus, and it's replication deficient. So it would not be able to replicate, but the immunogenic structures of the viral particle is completely kept intact. Just the genetic material has basically been replaced by the spike protein RNA code. The vaccine has already been approved just very recently last week in the United Kingdom and in Argentina. India followed suit yesterday. And AstraZeneca has conducted their trials quite differently. So they, in fact, started phase 1 trials and then moved on to phase 2 and 3 in the UK, and later also opened vaccine trials in Brazil and Argentina. And the data that were presented as an interim analysis were essentially pooled data from 3 single-blinded and 1 double-blinded placebo-controlled randomized trials. Interestingly, the placebo individuals did not just receive like a saline shot or so. They actually used an actual vaccine, Menactra. Menactra is a meningococcal vaccine, which is a very immunogenic vaccine on its own. Typically, it's very well known for producing a lot of vaccine local side effects like pain and swelling, inflammatory side effects. So this was a complete adult trial, 18 and above, and there were a few sort of hiccups in the trial which are noteworthy. The trial was halted in beginning of September. There was a lot of press about it after there was a case of transverse myelitis in one vaccine recipient. Transverse myelitis is a disease of the spinal cord where inflammation ensues to the point that individuals have neurologic symptoms which can range from various neurologic findings of numbness, tingling, all the way to complete hemiplegia, quadriplegia. Depends really how intense the problem is. And then there was a really funny and well sort of press uptake thing that occurred, that some of the vaccine recipients inadvertently only received half of the anticipated dose. And that was completely inadvertent and like a mistake, essentially, in terms of administration. So this is a major trial violation. But those individuals were still assessed just like in the intention to treat analysis. And we have separate data now and essentially these low dose administration versus high dose administration. And I'll show you the rather surprising results here. So the UK approval was based on these exact data. And here I want to point out, I'm going to take the laser pointer here. When you see here, this denotes low dose versus standard dose and first dose versus second dose administration of the vaccine. You see the numbers are much smaller compared to the Pfizer or the Moderna trials because it was only a small subset of individuals who received this low dose before the trial monitors noted that the vaccine administration was faulty. But interesting enough, the individuals who received the low-dose vaccine had a better response in sequence low-dose to high-dose or normal dose, standard dose, compared to the standard dose individuals. There was literally a 90% versus 60% efficacy in comparison. And that was statistically significant despite the much lower numbers, which is astonishing. And there's been various explanations rendered for this effect, where there's potentially even immune system priming that's better with just a low dose initially and a more vigorous response after a high dose administration later. Was there potentially immune system response against the high dose, meaning against the adenovirus particle that prevented a more proper immune response to the actual product, meaning the genetic product from the spike protein? We don't really know. And this is an interim analysis. We do not have complete data. The FDA has not reviewed this as of yet, and even the European administration that's essentially the correlate to the FDA and the European Union is not anticipated to have any word about approval until end of January. So rather astonishing.
I wanted, Dr. Pusch, if you don't mind, I probably should give you about a 5-minute warning.
Sure.
I do have quite a number of questions that have come in, maybe less so on the science side, but I just want to give you a heads up on the timing.
Sure. What's this about the vaccines? So duration of protection. There's a very robust antibody response against SARS-CoV-2 in regards to neutralizing and T-cell mediated cellular immune response. We know that from the phase 1 clinical trials. This is from the Moderna trial, where we have now about 3 to 4 months data, and people in phase 1 have sustained high levels of neutralizing antibody titers, which are more and more coming into the picture now as a correlate for sustained immune response, and it seems that's the case. So some FAQs, as I mentioned in the beginning. So the timing of the second dose, co-administration, etc., all of those things are addressed if you're not really sure where to go. This on the bottom is the hyperlink to the current CDC page. This page came online on December 30th. You will find a lot of answers to potential questions that we may not be able to address today. Patients with what's called Guillain-Barré syndrome, should they receive the mRNA vaccine? The unequivocal answer at this point in time is yes. This is concordant with the CDC guidance. There's no clinical cases in the clinical trials that have occurred, and as such, we do vaccinate people who've had a history of GBS. What about Bell's palsy? There was also in the lay press quite a bit of noise about it. Well, I can offer you these sort of statistical data answers. In the Moderna trial, there were 3 vaccine and 1 in the placebo recipient individuals who had Bell's palsy. In the BioNTech trial, it was 4 in the vaccine recipients only. It's still, if you match this out, this is the same as it is in terms of natural occurrences. This is per CDC data, which over the years has been stable at about 10 to about 30 cases per 100,000 population per year occur. Open questions. Prevention of asymptomatic infections, trial did not address. Sufficient protection in pregnancy, children, and less than immunosuppressed individuals, same scenario. They were excluded from the clinical trials. They have not been studied, and we really don't know. So what about prevention of asymptomatic infection? The trial did not address. I think I have a duplicate slide here. Should patients with anaphylaxis history to other vaccines receive the mRNA vaccines? Well, patients with such history were excluded in the Pfizer trial. As such, we don't really know, and the CDC's current stance is not to vaccinate individuals as such. What about prior COVID infection? Those were also excluded from the trial. So a small sample in the trial retrospectively were found and were vaccinated. The reactogenicity is not entirely clear. So at this time, it's also listed on that CDC page I just mentioned. Technically, individuals, once the COVID symptoms have resolved, can get vaccinated. The stance right now is that there's about a 3-month waiting period. After 90 days, an individual can be vaccinated. We know that the vaccine response in regards to measuring titers is higher if you compare neutralizing antibody titers compared to natural infection. How this relates into clinical response is completely unclear. But we do offer any prior COVID diseased individual the vaccine. What about the monoclonal antibodies that have received emergency use authorization recently? It's similar. We wait for about 3 months until those individuals will be offered vaccination. Should you take the vaccine? That's important to know, right? So I can offer my personal opinion here. The actual infection is likely to affect the person with no immunity to a far greater extent. The potential for complication from natural infection is far greater than from the vaccine, and the odds of having vaccine side effects is rather minuscule in comparison to the natural infection. So it's a very small calculated risk versus a very uncalculatable risk. And then altruistic behavior, which I mean is once you yourself receive the vaccine, you're much less likely to get infected and spread the infection later on. So for individuals who will either not take the vaccine or individuals who do not have a good vaccine response, you will protect them with creating or becoming part of the herd immunity. Yeah. So there's no doubt of what to do currently. Everybody should take the vaccine, no exceptions. I can stop at this point and hold discussions about the COVID vaccine strains.
Thank you, Dr. Pusch, and that actually will be one of the questions. Not unsurprisingly, some of the questions that I've received, have to do with distribution of the vaccine, timing as to when people might be able to expect to receive a vaccine. So the first question I have is, given the Oregon Health Authority has said that vulnerable individuals with chronic health conditions and adults over the age of 65 will be in the latter part of Tier 1, how will these individuals be identified and notified about their opportunities to receive the vaccine? That's especially important for individuals who live independently. In other words, they're not in a retirement, or a care facility. Could you address that?
Well, sort of, Kevin. See, I'm really on the medical treatment side for the most part, and I'm really not involved in the distribution at all. This is more a question for, OHA administrators, for our internal, Providence administrators. I have not been asked. I don't sit on any of the committees who decides, who looks directly at the logistics and gets this process going for the cold chains. So, I'm at the receiving end in that sense, just as everybody else is. So, yes, it makes sense to vaccinate everybody above age 65. It really is a logistical effort, and no, I can really not offer a great explanation or a timeline on that because I'm really not part of the decision, body.
So if another question, if you are called in, to receive a vaccine, do you have to be completely healthy when you come in? What if you've had a cold? What if you're experiencing a cold, for example? What if you fairly recently have had shingles as another example? I mean, are there any recent health issues that you would perhaps suggest where that person may need to defer receiving the vaccine?
Absolutely. So that's an important question. And we of course don't know details about this vaccine. It hasn't been tested out in co-administration with other vaccines, and we generally vaccinate people that feel otherwise well. That being said, low-grade temperature, having a cold is not a contraindication to virtually any vaccine that we give in the traveler's clinic. Could symptoms be potentiated if you already don't feel well? Yes, of course they could. Is it advisable to still move ahead if it's your turn and you would be able to get the vaccine? I think if it gets to a question, where you sort of lose your vaccine slot, that where you would receive the vaccine and you really want to take it, I would move ahead and get it. You delay essentially your own immune system generating a response to the vaccine and to a more normal life, if you will, and be part of the herd immunity. I think it's more of an esoteric question. How much risk are you willing to take? If you already have a headache and the vaccine may give you a headache in addition, of course, you might be feeling much more miserable than you normally would. But what we expect from the vaccine in general is very minimal side effects. So having a benign cold from a rhinovirus shouldn't really alter your decision to take the vaccine, yes or no. If you were just hospitalized with a significant illness, having pneumonia, I would probably wait until you're done with taking an antibiotic, taking other medications. You just may potentiate symptoms, nausea, throwing up, etc. So I think it's more a common sense decision than anything else. We have no signals from the vaccine to deter you really not taking it.
Another question, what are your expectations for how long the vaccine will last? And, are you anticipating that we'll need to be getting vaccinated each year, just like we do with the seasonal flu?
Yeah, super important question. And of course, nobody has an answer to that. I don't know, of course. From the phase 1 data that I just showed, people seem to have lasting neutralizing antibody titers at least 4 months in, or more than that. So the most recent paper that was published from the phase 1 study was beginning of December, and it quoted data that were assessed by October. So we're already 3 months further on now. I haven't heard any updates, but at that time, from the phase 1 trials from April and May, people still had sustained antibody responses. Could they suddenly tank and go down, and that would sort of like the influenza vaccine necessitate yet another injection on like a year-by-year basis, as you just quoted? That's possible. There's no human clinical trials that we could compare the mRNA vaccine to. For adenovirus vaccines, that's much better established. Could they last for 2-3 years? Yes, that's very well possible. Could this still be for, the adenovirus vaccine be a yearly vaccine? Yes, also possible. I'm hopeful, and looking so far at the antibody titers, I think it's reasonable to think actually that it lasts longer than a year. So yes, I think it will be a better vaccine than the flu vaccine, but we really don't know.
You touched on this a little bit, but, and again, we're going to speak in general terms because, of course, each person should really have this conversation with their primary care physician. But if an individual had a severe nut allergy, for example, to the extent where they've actually even gone into anaphylactic shock on several occasions, when they've eaten nuts, what's your response to a person who may be in that situation as it relates to getting the vaccine?
Yeah, and now that the vaccine has been administered, there have been a couple of cases of anaphylaxis in the UK with the Pfizer vaccine. It will occur. The incidence is extremely low. As I said, they were excluded from the trials primarily. So it's hard to know what to say in individuals where we exactly know that they have certain reactions of anaphylaxis, how they will do. Should they be pre-treated? Should they get, let's say, steroids, Benadryl, and get the vaccine in a really controlled setting, ambulatory, in a clinic, and have literally the epinephrine, syringe laying right next to them? I think it gets really down to an individual decision-making process. If it would be me, I would be willing to take the risk in a really, clean scenario. I would not go to, let's say, when it becomes available at Walgreens, go in there, get the vaccine in that kind of scenario. No. And that was exactly also the reason why the CDC initially said, no, this is a vaccine. We're going to start off and vaccinate only in the healthcare setting, in facilities, not in outside clinics, so we have the best possible response to people who have a severe side effect. So yeah, I think it's individual decision making there.
So another question, you were going to just actually talk about some of the variations that we're starting to see now in various countries. Do you have any sort of generalized concerns about these variations, and particularly as it relates to the effectiveness of the existing vaccines?
I have concerns about the virus in regards to, and I can move one slide forward here. This is the slide today from the New York Times that was published just this morning. And that shows the caseloads in Britain and in South Africa, where the variations are now the predominant strains. And you see, in comparison to the surrounding European continental countries and the surrounding African countries, that there's clearly an uptick in cases. And it's possible that the vaccine strain that's altered has the responsibility in that, but we have no knowledge if that's truly the case, because people's behavior in terms of social distancing, masking, etc. There's many factors that could explain this, so it's very biased. But this is what's being seen, and potentially this can happen. Am I concerned about the vaccine? Well, not offhand. Why is that? Well, you would have to have a mutation that would be significant enough to alter the binding in the spike protein sequence. That would have to be altered in order for the vaccine to not be effective anymore. That usually, with what we've seen so far about the genetic code and genetic alterations over the last year, is not likely to be the case, but we're looking at rather years rather than months, like influenza, where this kind of alteration will take place. So not likely to be clinically significant. The other thing you have to sort of take into consideration is that mass vaccination will take place very quickly. So if it would be a very slow, drawn-out process over the next 5 years, then probably vaccine escape mutants will develop, but I doubt that's going to be the case with coronavirus.
Another question has to do with your predictions for the future. If you were to look into your crystal ball and it's now September of 2021, can you just sort of talk about what you think the world looks like at that point? Are we, how much headway have we made against the virus, either in the U.S. or on a global basis? Are we returning to work? You can answer that however you'd like.
Yeah, I'm actually very optimistic about that because it's not just in the U.S. that we've made tremendous progress in regards to the vaccine development, but I showed, on slide 1, there's literally 100 different types of vaccines being developed in every single place on Earth. And those vaccines will be licensed. Most of those we will not see in the U.S., but they will be licensed locally and have their effectiveness. They may not be 95% like Pfizer, but they will have efficacy and create global herd immunity against the virus, and the numbers will drop significant enough that we will return to a semi-normal life. And it will really depend on us, on the logistics, and on us having a proper campaign for the vaccines against the virus so that a sufficient amount of the population will get vaccinated so that herd immunity is much better than 60% of what the kind of predictions is regards how many people will take the vaccine in this country. The more people will take it, the better the herd immunity is. There's various predictions of how much we need. We really don't know. It won't be like the measles, where you need 95% roughly in order to have herd immunity enough that outbreaks don't get perpetuated. For COVID, it's been anywhere between 60 to 80%. Probably the higher number is more realistic. So yeah, it's really up to us, public health campaigns, and the population, understanding how important this is, and get away from these, conspiracy theories and various concerns about vaccines in general. Yeah. But I'm looking very optimistically in the future.
Thank you. We need that. You know, I know several people asked questions that were sort of in that distribution vein of my initial question.
Right.
I will speak with the chief medical officer here at St. Vincent, you know, who's very in tune with, you know, the state's plans around those types of issues. And so I'll respond individually to those of you, you know, who posed those types of questions once I get some feedback from Dr. Moreno. But in the meantime here, in the few minutes that we have left, does anybody else have any questions that they'd like to pose? And just a reminder, if you'd like to speak, please just go up to your toolbar and click on the little microphone button so that we could hear you. Or you could put something in the chat box. All right, I'm not hearing anything. So Dr. Pusch, thank you very much. Very much appreciate your sharing your extensive knowledge here, you know, on the topic and for addressing some of the questions that have come up. And we appreciate all of you who took the time to join us today, and certainly wish everybody a happy and safe new year. Thanks for joining. Take care.
Thanks for having me, Kevin.
Thanks, Dr. Pusch.
Thank you very much.
Do you have to do it?